I think I added extra coffee grinds to my coffee today because I'm feeling really, woo, let's go. All right. So I'll try not to go too fast. Just give me a shout if I'm going a little bit too fast. So we left off talking about organelles and their functions. You should be familiar with the organelles of the cell. So we talked about last week, I know it was before your exam, so you might not be fresh on what we've talked about so far. We learned a little bit about the different types of cells on Earth. We compared prokaryotes and eukaryotes. We learned about cell theory, our modern understanding of how cells are built and operate. We started talking about the associations with cells. So we talked about the extracellular matrix and how important that is. From there, we moved to talk a little bit about the cell membrane and the fluid mosaic model that it exhibits. And we moved inside to talk about the components of the cell. So we left off talking about organelles and their functions. You should be familiar with those. And ooh, where are we on time? Maybe once I finish a couple slides, then I'll do another poll to kind of check where you are with your organelle understanding. We left off talking a little bit about energy conversions, really important in our cells. Remember that those energy conversions are referring to the chemical reactions that keep us alive. And the mitochondria, we're going to spend a lot of time in, as well as the chloroplast, learning a little bit about how ATP is cycled. I should say cycled because you're not making it from nothing. All right. In terms of more of the non-membrane bound organelles, we've got cilia and flagella, which both share this characteristic that they're oftentimes used in motility. You got to learn a little bit about cilia, tiny bit, in that Patterns in Science lecture assignment that I gave you. So we're going to talk a little bit more about cilia today. Cilia, as you got to see previously, right, are these small cellular extensions, right? They coat usually the surface of a unicellular organism. Cilia are much shorter in length than flagella, and they work in collaboration with many individual cilia, right? So you have a lot of cilia working in a coordinated fashion in order to create motion at some scale. They have a coordinated motion that's using ions, right, to coordinate. The way cilia work reminds me of the wave, right? You guys did a wave at the stadium, and how arms, right, go up and move in a coordinated fashion that makes this wave across the crowd of people. And if you've ever had that experience where somebody throws a beach ball out, right, and that beach ball rides that wave of moving arms. Cilia is the same thing at a small microscopic scale where those cilia are waving, right, just like arms in a stadium, in a coordinated fashion, right, that moves like a wave across the surface of the organism, or the tube that's lined with cilia if we're talking about a human body. And if you imagine that beach ball moving across the wave, you can now imagine how a female egg, right, moves down the oviduct using cilia to transport it along, right? So cilia, short, there's many of them, and they're working collaboratively to create motion. And that motion can be moving a unicellular organism, like you see in this little top animation. Looks like a couple little ciliates in there. And you can also use cilia at a larger scale. So for example, in the human body, we're going to have a few areas where tubes inside of our body are going to have cells that are lined, their surfaces are lined with cilia, and those cilia can help to move and transport substances through the tube. So in your respiratory tract, right, you've got cilia that are constantly moving mucus-laden debris up and out of your respiratory tract to keep it from making its way into your lungs. In the oviduct, you have cilia that are moving the egg after it's been ovulated. In the male reproductive system, in a funky part of the tube called the epididymis, right? The epididymis has cilia that are moving sperm around. So cilia used for transporting. Flagella, on the other hand, are much longer extensions, and they're going to be working in a more of an oar-like fashion, right? Kind of the way that hummingbirds' wings, right, move. And they're generally longer and fewer in number right? So we're mostly familiar with the way that we exhibit flagella in the human sperm, where there's one flagella. Indeed, flagella tend to be much less numerous, right? When you've got big whipping cellular projections, it turns out that they can start to disrupt their function. So generally we see 1 to a handful of cilia in most, flagella, pardon me, in most organisms that are flagellated or have a flagella. Does make sense? Working to propel that organism through the environment, right? So like a sperm moving through the female reproductive tract, or a unicellular organism moving through a pond of water. As you see this little euglenid here. Good? Cilia and flagella? Fun stuff. All right. Okay. So within eukaryotic organisms, we have 2 types of cells, right? So remember, there's 2 categories of cells on Earth, prokaryotes and eukaryotes. We're focusing in on eukaryotes. For eukaryotes, within a eukaryotic body, right, if we're talking about a multicellular organism, we sometimes see 2 types of cells, right? And in fact, even unicellular eukaryotes can produce sex cells. Actually, I should clarify that. All right, so the 2 types of cells that we'll see are somatic cells, and remember that that word root soma means body, right? So somatic cells are literally body cells. Any cell that you're imagining other than egg or sperm is a somatic cell. So most of the cells in our body are somatic, and they're gonna, as we mentioned, be very diverse in their shape, right? And their functionality depending on where they are. And we're going to see that somatic cells are going to replicate themselves using mitosis, right? So it's a type of cell division that makes duplicate identical copies of cells versus your sex cells, right? And sex cells are important for sexual reproduction. I'm going to talk a lot more about these when we get to cell division. And I will also talk about them when we talk about reproductive system. So sex cells are only created in organs called the gonads, and that's a generic term for a reproductive organ. It refers to either an ovary or a testes. Keep in mind that the ovaries and the testes themselves are made up of somatic cells. Inside of them, they can create, excuse me, sex cells. Right, so sex cells are very unique types of cells in a eukaryotic body in that they contain half of your genetic material, and the way that they're created is by a process called meiosis. Again, we're going to come back and talk about mitosis and meiosis later in this unit, so it'll all amalgamate as we get towards approaching the exam 2, right? You'll be able to tie those 2 concepts together. For now, let's leave it at, I've mentioned mitosis, meiosis, so they're not these foreign concepts, but we'll come back and learn more details about them in a week or so. Any questions on the 2 types of cells in a eukaryote? Okay. We also oftentimes will talk about plant and animal cells, and I think this is kind of one of those historical concepts that's added into intro biotechs, and I'm not super keen on it because this list of here are the characteristics listed in your book that only animals have, and here are the characteristics that only plants have, as if it's that simple. It's not, right? Almost all plants will have chloroplasts. They will definitely all have that cellulose cell wall. But we do see that some plants, especially ancient plants like mosses and ferns and cycads, still have flagellated sex cells. So this list kind of breaks down. It's not universal. A couple of the things that animal cells have that most plants do not is centriole, which we're going to learn is very important for cell division. I'll talk about that soonish. And then that lysosome that we talked about, that structure or organelle that is designated for intracellular digestion inside the cell. So I do want to point out one thing, one misconception, and really if we wanted to talk about diversity of cells, we would add fungi in here, right? Because fungi have really crazy cell differences as well. But if we take a look, this is the one misconception that I want to make sure that we understand. Remember that a cell wall is not the same as a cell membrane. They're two different things, right? The cell membrane here is illustrated in yellow. It's that phospholipid bilayer with the mosaic of proteins and cholesterol and other molecules in there. I want you to notice that that yellow is still here in a plant. A plant still has a cell membrane made out of phospholipid bilayer, right? It has, in addition to that, this outer wall made up of cellulose, right? That is the key difference that I guess I want to project here. Does this make sense? Yeah. Okay. All right. So what I'm going to spend the rest of lecture today talking about is cell function. Right? Cell function is looking at all the things that happen inside of the cell. For today, we're going to be talking about water balance and movement within a cell. And then as we progress through this unit, we'll learn about other cell functions like division and gene expression and other fun stuff that's happening in there. But water transport and water balance is really critical, right? Living organisms evolved in water, and you had to figure out a way to regulate your water balance. And not only do you use it for maintaining homeostasis, constant internal body conditions, you can use water movement for a bunch of different functions, right? So here what we're looking at are these fruits being produced by this plant, and they build up water pressure, and then when the stem breaks, that water pressure whoosh, releases and disperses seeds, right? So movement of water can be used for all kinds of cool things, right? In both plants, animals, fungi, protist groups. So just to recap what we've already learned about movement in a cell, we talked a little bit about membrane permeability, how things move across the plasma membrane. So we said because of this hydrophobic zone right in here, right, where the tails are in your phospholipid, that hydrophobic zone makes it really challenging for polar molecules to move through, right? Because remember that polar molecules are hydrophilic, right? And they're going to have a hard time making their way through this nonpolar hydrophobic area, right? I always imagine they're going to bang around in there like a ping pong ball, right? Unable to move through readily. So what we're going to see is that nonpolar molecules are going to have a much more easier time making their way through this nonpolar zone, right? So they're going to diffuse and be able to move through in between the phospholipids and across the plasma membrane, in or out of the cell. We call those things lipid soluble, right? Because they are interacting well with other nonpolar molecules, right, or lipids, which are nonpolar. So lipid soluble molecules, like something like maybe testosterone, right, might move through readily, and that's indeed a really important chemical hormone messenger that regulates gene expression in our cells. Small also is going to move through readily, and conversely, we're going to see that large molecules or really bulky branch molecules are going to have a hard time moving through the phospholipid membrane. And for that, we mentioned that we would use transport channels for polar items, for large items, for bulky items. We're going to tend to use transport channels, right, or some type of membrane protein that's going to help us to move those substances across the plasma membrane. So moving things in and out of your cells is collectively referred to membrane transport. And actually, I forgot, I was going to give you another poll. Let me. I spelled organelles wrong. The keys are still sticking. I have purchased like fancy vacuums and pokey things for cleaning out the keyboard, and nothing seems to resolve it.
The post just like disappeared for me. It showed up and it just disappeared.
Okay, you're gonna have to email me because I don't have a way of relaunching it without starting it all over for everyone.
Okay. So that's the second post, right? The second discussion.
Mhm.
Okay.
So the site of protein synthesis. Vote. We're getting there. All right, so while you guys are finishing up your voting, I'm going to keep talking. So membrane transport, transporting things across the plasma membrane can be expensive, right? Or it can be cheaper in terms of energetic costs, depending on the type of transport that we're utilizing. So this is what we're going to spend the next chunk of time talking about is what's the difference between passive and active transport. All right, so active transport to start off with, I'm going to talk a little bit later today, is any type of transport that requires a direct investment of ATP energy. You have to put in ATP in order to be able to get across that plasma membrane. So it's kind of, imagine it like paying a toll, right? You have to pay that ATP energy in order to be able to move product across. And this is generally when we're moving a lot of a product across at any one given time, right? So we're going to talk about bulk transport and the mechanisms that we use for bulk transport, something called endocytosis and exocytosis. And then we're going to be learning in a minute about concentration gradients, and we're going to learn that molecules like to move from an area or have a tendency to move from an area of high concentration to low concentration, right? From an area where they're crowded to an area where they're dispersed. And when we want to move them in the opposite direction from an area where they're dispersed to where they're crowded, right? It's going to require energy to force them to move in that direction, right? So I'll come back and talk about those two in a bit. All right, last call for voting. Active transport. All right, so looks like you're doing okay-ish with understanding the functions of your different organelles, right? This is a great set of material, the organelles and their functions, to put on flashcards, right? So you can quiz yourself. Indeed, most of you are on the right track. About 66% of you voted for ribosomes. Indeed, it is the site of protein synthesis. Ribosomes are going to help to coordinate the dehydration synthesis amongst amino acids as you're building that long polymer. So go back and review your organelles if you're still struggling with what they do. Right? If this is the first time you're hearing this, right, it takes a little bit longer to process that information. All right, let's put that away. Okay, back to transport. So active transport, we just talked about, requires a direct investment of ATP energy. For passive transport, you're not using a direct investment of ATP energy, right? Sometimes you might use indirect investment, but mostly you're not using ATP to fuel this type of motion. So we're going to be learning about diffusion, osmosis, filtration, and facilitated diffusion. I'm going to spend most of my time on the top two, right? And then I'll just mention the bottom two for completeness. For each, we're going to talk about the ways that they work, the types of molecules that they work for, and we're also going to talk about what is the energy source that's being used. One of the things that we're going to learn in metabolism, our next lecture, is that when you're moving things, that's a type of work, right? And work requires energy. And if it's not ATP energy that you're paying, it's some other energy source that you're going to be using to move these molecules around. So we'll talk about what are some of those energy sources. First type of passive transport is diffusion. And diffusion is a concept that you're going to want to understand really, really well. Right. So diffusion is going to be a process of moving molecules or atoms around from an area where they're high concentration to an area where they're in low concentration. In other words, this is what we call moving those molecules or atoms down their concentration gradient. And if you've taken chemistry already, you know that these brackets, right, mean concentration. Right, so we can say we're going to move molecules from an area of high concentration. You want to come under the table? To an area of low concentration. Right. When molecules are moving from an area of high concentration to an area of low concentration, we refer to this as moving down their concentration gradient. Right. This is the natural tendency of molecular movement by diffusion. From high concentration to low concentration. And intuitively, right, we all like to go from an area of the cafe where it's crowded, right, to an area where there's more space, right? We all like to move from crowded to more dispersed. And there's going to be a lot of things that are contributing to diffusion rates, right? Let me go back for a second, right? And show you the animation, right? So here you can see how the molecules are going from an area of high concentration and dispersing until they're evenly dispersed in their environment. Areas of low concentration. Now, here's what's driving this. You guys, have we talked about mosh pit? Right? I think we did talk about mosh pit. Remember we said mosh pit, all the atoms in a mosh pit are bumping into each other, right? And as they bump into each other, they get dispersed away, right? Just how you get, boom, thrown off the edge of the mosh pit. Right? So molecules, when they're in an area of high concentration, are going to be bumping into each other. And remember that they're have that kinetic energy, the energy associated with molecular motion, right? Molecules in the environment are going to be moving around. We talked about this when we learned about heat, right? And as they bump into each other, they're going to move away from each other. So of course, in an area where those molecules are crowded, they're going to be bumping into each other more frequently and dispersing themselves, right? They're naturally going to go from areas of high crowdedness, high concentration, to areas of low concentration, right? Until they're evenly dispersed, right? It's not a directional movement, it's random, and it's using just that molecular motion, that atomic motion, right? The kinetic energy associated with that. It's going to be the energy source. So what's going to impact diffusion? How concentrated you are. The more concentrated you are, the more atoms are in there to bounce around and disperse. So the higher the concentration, the faster the diffusion rate. What else do you think might speed up diffusion rates? Those molecules.
Temperature.
Temperature, right? The more agitated they are, the more frequently they're going to bump into each other, right? And disperse themselves in the environment. So you know a lot about diffusion already, right? If you are on a road trip, right, and somebody in the back seat has stinky feet, right, and they're trapped in the stinky shoe. Right? As soon as you take the stinky shoe off, right, there's high concentration of stinky feet molecules in that shoe. And what's going to happen? They're going to start diffusing, right? Because space is opened up for them. And they're going to diffuse all through the car until they're equally distributed. And everybody can smell the stinky feet, right? That's diffusion. Yeah? Okay, so diffusion can happen across a plasma membrane. And here's where we can add some more complexity. This makes it a little bit mind-boggling to think about. So diffusion will happen naturally across a plasma membrane, provided that that molecule is permeable to that membrane, which means it can get across the membrane. There's a membrane protein for it, right? Or it's small and lipid soluble and can move through the plasma membrane. And what we're going to do is we're going to see that those molecules are still going to move. So in this case, we're looking at these purple molecules. This band in the middle represents the plasma membrane. And notice how the membrane is permeable to the purple dots, right, to the molecule. So they're going to be able to move across the membrane. And they're going to be moving from the side of the membrane where they're in high concentration to where they're low concentration. And they will continue to exhibit a net movement until they're equal concentration on both sides of the membrane. We call this equilibrium. Now, here's the thing. They're dynamic equilibrium. Because remember, this is random motion. The molecules aren't saying, hey guys, let's go over there. Right? They're just randomly bumping into each other. And where they're bumping into each other, where they're more concentrated, they're going to bump into each other more frequently and disperse naturally to the side where they're less concentrated. And what I want you to look at this animation, you'll see that there's a net, an overall movement from the left side where there's high concentration to the right side where there's low concentration, the net movement. But if you watch carefully, you'll see that while most molecules are moving to the right, there are still some randos moving to the left. Because as they start to bump into each other over here on this side, some of them might get bounced back to the left side, right? It's a random motion. Where you're in higher concentration, you're going to have more molecules moving across the membrane to the side that's low concentration. but you're also going to have some randos moving in the opposite direction, right? So we refer to this as a net movement, right? Overall, it's happening down the concentration gradient. And then when the concentrations are about equitable or in equilibrium on either side of the membrane, the molecules don't all say freeze and stop. Right? They're still bouncing into each other. You're still going to see some randos going to the left, and you're going to see some randos going to the right. Right? But they're going to be moving at an equal rate. And so the concentration equilibrium will hold. But it's dynamic because you're still seeing individuals moving. Does that make sense? Dynamic equilibrium. And this is only going to happen if the membrane is permeable to that molecule. Good? All right, easy parts. All right, so diffusion refers to this type of motion for any type of molecule, whether it's a hormone or an ion or stinky shoes molecule, right? Stinky feet molecule. Usually when we're talking about a solvent, the dissolving agent, you guys remember that term, solvent? We're looking at diffusion of a solvent is called osmosis. And remember that the universal solvent is water. And so we oftentimes will hear that osmosis is the diffusion of water. It's technically the diffusion of any solvent. But in biological systems, we're going to be talking about the diffusion of water because it's our universal solvent. All right. Well, in this case, we're going to be looking at water molecules. And water molecules are going to be moving from areas of high concentration to areas of low concentration. Right? And if the membrane's permeable to that water molecule, the water, if you watch the animation, the molecules are going to, little blue water molecules are going to start off high concentration on the left side and move down the concentration to the right side. You guys see it? Okay. Same thing, random molecular motion is going to be driving until you reach dynamic equilibrium. Yeah? Okay, are you ready to add the part that makes it confusing? Ooh, that stopped my animation, yay. But not in the right part. It'll work. All right, so here's the funny thing, right? Water is always going to diffuse down its own concentration gradient. Any molecule is going to diffuse down its own concentration gradient, right? And indeed, we can see water in high concentration on the left. It's going to be net motion moving to the right. But oftentimes, when we're talking about osmosis, we don't just look at the water molecule, we look at the overall solution, right? So here, if we take a look, I'm going to change this to blue really quick. I'm going to use blue for the water, right? The water here is in high concentration, and it's going to be moving over here to where it's in low concentration. That we've already talked about, right? You see a net movement in this direction. What I want to talk about next is the solute, as the solution, pardon me. One color, let me go back to red. If we take a look at the solution, the mixture of the solute here in green and the water in blue, the solution, what do you guys think? High or low concentration here?
Higher.
So you have a little bit of solute relative to solvent.
Oh, lower.
Lower. Right? So a little bit of Gatorade powder relative to the amount of water. And over here, you've got a lot of solute relative to the solvent. You've got high concentration. Yeah. And this is of the solution. Oops. It's supposed to be a little apostrophe. Yeah. Now, the membrane is not permeable to the solute, but we can use the solute to help manipulate water. Anywhere, what this means is that anywhere you have a low concentration of the solution, you're going to have a high concentration of water. Anywhere where you have a high concentration of the solution, you're going to have a low concentration of water. Does that make sense? They're kind of inversely related to each other, or they are fully inversely related to each other. And it's interesting, right? Because we can take advantage of this to manipulate water. What are you reading? It's fine. He knows when he's in trouble, he freezes. Hey! No. All right, so you can use this relationship to manipulate the movement of water, right? So notice how water is moving from an area of low concentration of the solution to high, right? We sometimes refer to this as, oh, water's following the solute. Water's going to diffuse at its own concentration gradient, but you can manipulate the overall concentration of the solution to cause water to move, right? So you can add a ton of solute to one side of the membrane, and that's going to influence the concentration, relative concentration of water. Right? So I guess what I'm getting at here, it's not trying to confuse you, but what we're going to see in biological systems is you can pump solute into one side of the membrane, and that's going to cause water to diffuse towards that side of the membrane because you're manipulating the concentration of the solution in order to encourage water to move in a given direction. Yeah? That one's a kind of a stretch to understand. So maybe I have a blood vessel, right? Maybe this side over here is my extracellular matrix, and I have a blood vessel delivering or diffusing from high concentration to low concentration because I just ate a sugary meal, right? That means when my bloodstream absorbs that meal, I'm going to have lots of high concentration of sugar in my blood vessels, right? So my solute, in this case, the sugar, is going to diffuse down its concentration gradient. If it's permeable to the capillary bed, which it is, it's going to diffuse into the extracellular matrix. Dump a bunch of sugar in here. And what's going to, or salt, if I had salty meal, right? And what's going to happen to the water from my cells as I dump lots of solute into my extracellular matrix? My water is going to move from my cells to the extracellular matrix by osmosis, following that solute, and I'm going to get all bloated, swollen. Right. Osmosis and diffusion are really important for how biological systems work. So you're constantly going to have to be working on maintaining water balance so that you can keep equal concentration of water in your cells and outside your cells. Right? And for that, to understand that, we're going to talk about osmoregulation. Any questions about diffusion or osmosis?
Yeah, I have a question about osmosis.
Mm-hmm.
Can you still stimulate movement of water by introducing more solvent or more solute if the water is at equilibrium? Like if it's balanced on both sides, will its concentration increase more on one side if there's more solute there?
Yes. And so what I've been telling you is that each molecule is going to diffuse down its own concentration gradient. This is true. But there's some complexity there in terms of like charge and polarity and concentration that also influences movement of molecules at the cellular level. And if you go on to take physiology, you'll get to do all the mathematical calculations, right? Of how things will move according to ion concentrations and all the nitty-gritty detail. So it's much more. I'm summing it down, even though it's more complicated, right? Just to kind of get you the gist of why it's important, right, to not be eating super salty, super sugary meals is because it's going to disrupt osmosis. Great
Okay.
question. Great catch.
Thank you.
It's complicated. All right. So osmoregulation is basically looking at water balance. And remember that water balance is incredibly important for all life on Earth, whether you're unicellular or multicellular. If you're living in fresh water, right, your concentration gradients inside of your body are going to be higher than the environment. Water's going to rush into your body. If you're living in salt water, right, the concentration of solute is going to be higher in your environment than in your tissues. And so water is going to constantly be rushing out. So organisms that evolved in water, whether fresh or salt, are constantly having to, adjust for that natural movement of water by osmosis, right? To maintain appropriate water balance inside of your tissues or your cell. This is something called osmoregulation, and osmoregulation had to evolve really early on, right? And one of the ways that, again, we can manipulate water movement is by manipulating solutes, right? So we're going to talk a little bit about the manipulation of tonicity. And tonicity is looking at the relationship between the concentration of the solution outside your cells relative to inside your cells. And as we just learned, right, that's going to influence the way that water moves, which is why I went into it, right? So we're going to talk about what's isotonic, what's hypotonic, what's hypertonic, right? So this is one where you want to really put your thinking caps on because this is one of the harder concepts, I think, for folks. So I don't have a chalkboard, so I'm going to just use this animation. I'm going to start off with isotonic, right? Iso means the same, tonic is solution, right? So isotonic solution is basically what it, right? But in any case, isotonic solution, iso means the same, right? So you have the same concentration outside of the cell that you do inside of the cell. Right? And that means that your cells are in dynamic equilibrium, right? You've got no net movement of water in either direction. Some's going in and some's going out. Isotonic solution is where you want to have, isotonic conditions is where you want to have your bloodstream all the time, right? In isotonic conditions. Your extracellular matrix, for the most part, you want to be isotonic, right, with what's happening inside of your cells. In animal bodies, right, cells want to be in an isotonic solution if they're regulating water balance, and there's a few exceptions. Some marine animals are conformers, and I'm not going to get into that. Take my organismal biology class if you want to learn more about that. Sorry, I don't know what's going on today. Isotonic solution makes sense. Concentrations equal outside relative to inside. You're going to be in dynamic equilibrium. You're going to have a few water molecules moving out, but at the same rate that they're moving in. Animal cells are happy as can be here. Right. Now, what happens if we look over here, right? In a hypertonic solution, I always imagine hyper kid, lots of energy. Right? In a hypertonic solution, that means that the concentration of the solution, maybe I'll draw them. The concentration of the solution on the outside, ooh, it messed up my animation, is high, right? The concentration in the cell is low, right? Which means that water is going to be moving, right? I guess I shouldn't have done that, from the cell out to where the solution's in higher concentration. And what's going to happen in that process is as your water is moving out of your cells into the extracellular matrix or into the fluid of your blood, this is illustrated here, that cell's going to shrivel up. We call that, the fancy name for that called cremation or shrivel. Right? And that can kill your red blood cells. Can I make a hypertonic solution in my bloodstream? The fluid relative to the cells that are floating around in it? Every time I eat a really salty meal or a really sugary meal, right? all those ions or sugars or solutes are moving into your bloodstream as they're being picked up from your digestive system. And they make your blood immediately hypertonic. This is why blood from your digestive system immediately goes to your liver, where your liver filters out the excess solutes from the solution to restore isotonic conditions. Does that make sense? What happens if your liver isn't able to pull those extra sugars out of your bloodstream and they stay? What happens if your cells of the rest of your body aren't able to pull that sugar? Well, that's a different condition, but water is going to be moving into your blood vessels. It'll lead to high blood pressure. Disruption of sugar balance, disruption of your sugar concentration gradients. This is what diabetes is. Right? You can't regulate your sugar appropriately, and it disrupts concentration gradients all across your body. This is why diabetes is such a destructive disease. It affects all of your organ systems because it disrupts concentration gradients all throughout. All right, so let's take a look at the, any questions, hypertonic? Right, so we're talking about your bloodstream, but then those solutes will move down their concentration out of your bloodstream to your extracellular matrix and cause problems that we talked about before. All right, so let's move on and learn a little bit about hypotonic. And hypo means below, right? So like a hypodermic needle goes below your skin, right? Hypo. And hypotonic solution basically means that the concentration of the solution outside of the cell is low relative to the concentration of the solution inside of the cell. And as a consequence, what's going to happen is you're going to see a net movement of water in. Right? And the cell is going to start to swell and swell and swell until it ruptures. And when you kill your red blood cells in your bloodstream, say, X is in your eyes. Can you think of any way that we would make a hypotonic solution in our bodies? In our bloodstream?
Maybe if you drink too much water.
If you drink water really fast, like a lot of water really fast. Remember that your kidneys are also constantly filtering your bloodstream to restore it to isotonic conditions. Right? But if you're drinking water at a faster rate than your kidneys can pull it out, you end up making a hypotonic solution. Right? And indeed, that can be deadly. We sometimes call this water poisoning. Right? So the famous example I always talk about is there was a radio competition, right? Radio show, radio program competition, right? Where the DJs were like, okay, let's see who can drink the most amount of water in the time of our show, right? And have this big competition for, I don't know, concert tickets or something. And, you know, nurses were calling and saying, hey, that's really dangerous. You shouldn't be doing that. Hey, that's really dangerous. And they were like, it's just water, no problem, right? This is what happens when you don't take a biology class. And what happens is as you drink that water really fast and it moves into your bloodstream, it creates a hypotonic condition. And it turns out that they killed somebody by having this drinking water competition. Now, you wouldn't do something silly like that because you've learned all about this in biology class. Well, maybe not on purpose, right? But we see this sometimes happens when we're exercising a lot, right? And we're keeping hydrated, and you're drinking a lot of water, it turns out you're losing solutes as well. And so the loss of that water that you're sweating and the loss of solutes that you're using to move that water out of your body can result in hypotonic solutions. So this is generally, we see this in like, the high school football team was out practicing all day on a hot day and the kid collapses, right? But he's been drinking water. Been drinking tons of water, but not replacing the solutes, right? We see this also in marathon runners, right? We sometimes see them collapse because historically we'd have little stations with water, and they were drinking lots of water and creating hypotonic conditions. This is why now it's more popular to have like the hydrating goo, right? That's also replacing solutes in addition to the water. Make sense? All right, okay. Any questions on tonicity so far? Okay. All right, so if we talk about animal cells, as we've been mentioning, right? Animal cells like isotonic conditions, right? In hypotonic conditions, water and net movement of water, right? There's always going to be, all these illustrations show like the net movement. Remember, there's randos moving in the opposite direction. Hypotonic is going to cause the cells to rupture. Hypertonic is going to cause the cells to shrivel. Isotonic keeps them just right, Goldilocks. It's not true of plant cells. Right? Plant cells also have, right, this rigid cellulose wall. And so what happens is, remember that cell wall is important for structure. If you stack one cell on top of another, right, it gives you nice structure to your plant. But if the cell isn't really full of water, what we call turgid, right, really full of water, it's like trying to stack empty boxes on, boxes with just a little bit of weight in there, right? They're not really going to hold their structure well. So in hypertonic conditions, you're going to see the same thing. The cell's going to undergo crenation in a plant cell. We call it plasmalization. In isotonic conditions, the cell's okay, right? But it's not pushing up against the cellulose cell wall. So this is when your plant's getting a little bit wilty. It's not very perky, right? Plants are unique in that they like hypotonic conditions where water rushes in and causes the cellulose wall to really swell and be really, really nice and structured and stable, right? And in an animal cell, water would rush in until it burst, kind of like overfilling a water balloon. But the plant cell has this cell wall made out of cellulose that pushes back, it exerts pressure onto the plasma membrane and prevents it from filling so much that it ruptures. So the difference of having that cell wall is actually kind of important for plant cells in terms of their structure. So if we take a look, here's a plant on the left under hypotonic conditions, all the cells are turgid and filled with water, right? Versus hypertonic. Isotonic would be a little bit less extreme, still wilty, but not to the point where it's damaged the tissue. Yeah? All right. All right. So as that plant is able to restore water, if it's a little bit, here's isotonic condition, it can come back. Hypertonic, usually the cell's plasmalized, and so it's damaged for good. But plants manipulating, right, how much water they have in their cells can actually be how plants move water, right? Move. So here we're looking at what's called a stomata, and I'll talk about this a little bit more later on when we get to photosynthesis. But these are specialized cells that are constantly manipulating the solute movement so that they can encourage water to either enter in or out. And depending on whether they're turgid or not, it's going to cause this pore, this opening, to open or close, right? So they're manipulating solutes all the time to manipulate the movement of water to open or close these pores. These pores are on the underside of all the plant leaves. It's how the plant is regulating gas exchange as it's exchanging with the environment. You can also use the same principle to either fill more or fill less water into your cells. And as you do so, here you're looking at clover or the genus Oxalis here. And these guys are going to open up during the day to harvest as much sunlight as possible. And then they're going to change their turgidity to close up their leaves during the shady time of the day, a cloudy day, or at night. Right? So plants can use this for motion. And then here on the right, you're looking at a tendril, right? So this is a structure that a plant uses to kind of wind and attach itself. And the way that it's causing this motion is by differentially moving water into cells on one side of the stem versus the other, right? So manipulating tonicity can be used not just for homeostasis, regulating water balance, but plants can start to utilize it for different jobs, right? Like dispersal of seeds that we saw earlier, or movement. That makes sense? Lots of important applications to water balance. All right, so far we've talked about 2 types of passive transport, diffusion and osmosis. Remember, we have 4 types of passive transport. We also have filtration and facilitated diffusion. So we'll talk about those next. And again, I'm not going to cover them in as much detail, but I do want to give you a little bit of completeness of rounding out this idea of passive transport. So filtration is going to be a way of moving molecules as well in your body. And generally for filtration, you're going to be moving molecules across a semi-permeable membrane. And so the example that I'm going to talk about is how you use filtration in your kidneys, right? So in your kidneys, you've got these small specialized capillary beds, right? That their walls are built like a set of colanders, 3 sizes of sieves, right? So there's physical holes, right, that are present. And as the energy source that you're going to use, blood pressure, pushes blood through those porous capillary beds, any molecule that's small enough to fit through the membrane pores is going to move through and get filtered out of your bloodstream, right? So here's the bloodstream, right, on the right. Here's the membrane, the cell membrane wall of those capillary beds, right? And a hypothetical little pores, right? And there's 3 levels of sieves, and I'm not going to get into the structure of the kidney. It's really kind of fascinating, but take anatomy or physiology and you'll learn all about kidney function. Is really amazing. All right, so anything that fits through those pores is going to be pushed through as the blood is forcing, creating pressure, right? That's going to push anything that fits through the pores through, right? And that's going to get trapped and removed out of your bloodstream as filtrate, or what we call urine, once it's no longer being manipulated. Does it make sense? Filtration. The pressure is the driving force, the energy source in this case, right? And it's all size-based. So just acting just like a colander in your kitchen would. Filtration. Cool? All right. And then last category is facilitated diffusion. And it's diffusion, right? You're still using molecular motion as your energy source. You're still moving from a high concentration to a low concentration. You can still only move across a plasma membrane if the membrane is permeable to that molecule. The difference here is that instead of using a channel, a membrane protein that's just a doorway, right? It's using a different type of membrane protein that actually interacts with the solute that you're moving across. So as the solute interacts with the membrane protein, notice how the membrane protein changes shape, right? To shuttle that molecule across. It's facilitating or helping the process of diffusion across the plasma membrane. Doesn't require ATP energy. It's using kinetic action, and then sometimes when molecules interact with each other or form a little bit of a bond, it causes the molecules to change shape. We call this a conformational shape change. Right? So as the solute interacts with the membrane protein, it causes a shape change that helps to shuttle the molecule across. Good? It's diffusion. You're just now using the membrane protein to help. So it's facilitated diffusion. And that's as much detail as I'm going to get into that. Make sense? 4 types of passive transport. Should you know them each? Be able to describe them, right? Give an example. Know their energy source. Yes. All right. So we'll loop back around to talk about active transport next. Right? So active transport, again, requires a direct investment of ATP energy. Right? And we use it usually in two cases that we're going to talk about, right? Or the two primary cases that we use active transport for membrane transportation. Bulk transport, which we mentioned before, is moving a lot of a product across the plasma membrane at one time. So up to now, we've been moving one molecule or one ion at a time. We're going to talk about moving a bunch across. And then also, we talked about how active transport, you're going to have to spend energy to move molecules against their concentration gradient. Remember that a molecule naturally wants to move by diffusion from high concentration to low concentration. Active transport is going to be when you're using ATP energy to move molecules in the opposite direction that they want to go, right? So in this case, here you can see your plasma membrane. There's high concentration of your orange solute. Notice how it's in lower concentration here. We're having to invest ATP energy to pump, right, that solute from an area of low concentration to an area of high concentration against its concentration gradient. Takes energy. Those molecules do not want to move in that direction, right? And this is a concept we're going to talk about a lot, right, in the next couple lectures. We're going to be looking at the movement of H+ ions, right? And in our bodies, right, we're going to use ATP energy to pump hydrogen from areas of low concentration across a plasma membrane to areas of high concentration, right? This is going to be a really important way that we can store energy in our body by building a concentration gradient. And then use it later on. And I'll talk more about that next lecture. Let's wrap up active transport and talk about bulk transport. So for bulk transport, we're moving a lot of a product across the plasma membrane at one time. There's two types of bulk transport that we're going to talk about: endocytosis and exocytosis. So cyto, remember, means cell. Endo means inside of, right? So endocytosis is bringing a lot of a product from the extracellular matrix into the cell. Exocytosis is exiting, right? So dumping from the cell out into the extracellular matrix. So for this, you're going to use a vacuole, right? Remember, a small sac of phospholipids. And what's going to happen is that vacuole is going to fuse its phospholipids and incorporate it with the plasma membrane. And in that process, dump a lot of whatever it's carrying product from one side of the membrane to the opposite. And you're going to, depending on which direction this is happening, you're going to be either bringing it in, endocytosis, or dumping it out, exocytosis of the cell. So endocytosis, there's two types of endocytosis, phagocytosis or pinocytosis. Right? So phagocytosis is how you bring in a lot of something to the cell, right? So maybe you're bringing in a vacuole's worth of content or a large item. In this case, you're maybe bringing in a bacterial cell. You're bringing in a whole cell into your cell to attack, right? So this is probably representing something like a white blood cell. Goes around the environment, and endocytosis, or specifically phagocytosis, is a bacteria that's maybe pathogenic in your body. So bringing in a lot of a product or a large product, you use phagocytosis. It's described as cellular eating, and that's because in a unicellular organism made out of one cell, this is how they eat. Make sense? In our bodies, this is how our white blood cells engulf pathogenic cells or substances. Pinocytosis is also bringing something into your cell, right? But in this case, it's a solution. It's a mixture of solute and solvent, right? So it's oftentimes described as cellular drinking, right? Because you're bringing in a solution, you're bringing in a liquid, right? So phagocytosis bringing in a solid, pinocytosis bringing in a solution or a liquid, both bringing into the cell. And again, important for unicellular organisms for eating and drinking. Exocytosis is how we dump product, a lot of product out of our cell. So your endomembrane system that's transporting product out is going to be using exocytosis, if you recall. It's, I guess, if endocytosis is cellular eating, then exocytosis might be like cellular pooping. If you're a unicellular organism, it can be a mechanism that you use to eliminate waste, dumping it into the environment. In our bodies, it's how cells that are producing product transport that product out of the cell. So if I'm a salivary gland cell, I'm going to be exocytosing salivary amylase, the enzyme that I made for breaking down starches, right? Dumping it into the duct of the gland so that it can drain into my mouth. If I'm a pancreatic cell producing enzymes, I'm going to do the same thing. I'm going to be using exocytosis to dump my product into the duct and drain it into the digestive system in that case. So it's how cells move a lot of a product at once. Good? Bulk transport, moving a lot of a substance across the plasma membrane at one time. And then again, if you're a unicellular organism, this can be really important for how you eat or drink. So here you can see an amoeba. Endocytosis brings in a vacuole by pinching off the plasma membrane, eating two little ciliates called paramecium. And there you can see they're now trapped inside of the body. And as this unicellular organism fuses that food vacuole with lysosome vacuole that contains digestive enzymes, it kills the ciliates. Yeah? Amoebas. Ooh, I've got a few minutes left. Let's start metabolism. Any questions? Cell function?
I have a question.
Yes.
So for the exocytosis, so both the like the liquid and those like the solid things just go out just one form, not like when they bring in separate like different two forms, right?
Yeah, correct.
I have a question really quick, Professor.
Yes.
So for exocytosis, when the cells are dumping the large product, is that also using a vacuole?
Yeah.
Okay.
So you have a product generally, remember we talked about endomembrane system and how it uses transport vesicles?
Yeah.
Those vacuoles for transporting? Yeah. So you're using a transport vesicle that's fusing to the plasma membrane.
Okay. Awesome. Thank you.
Vacuoles are really important for endocytosis and exocytosis. You're always going to be using vacuoles of one sort or another. Great questions today. I love it. Anything else? I can't for the life of me get the times down. There's something about the working from home, Zoom, timing. I have 5 minutes with you guys. I've been trying to remind myself, okay, at the 45-minute mark, that's when I have to let them out because I've been keeping you guys late too frequently because I keep losing track of time. So yesterday in my Monday, Wednesday class, it's out at 1:55. I'm like, okay, the 45-minute mark. Okay, see you guys. Sorry, I kept you late. And I let them out 10 minutes early. It's like, I wanted to put sticky notes up. All right, so the next PowerPoint I have, I'm going to just introduce energy and energy uses so we can start talking a little bit about how we cycle energy in the body, right? So what we call metabolism, right, is basically cycling energy in our bodies to fuel our activities. So I'm going to talk a little bit about work in the next 5 minutes. This is a short PowerPoint, so it's just to introduce some of the generic metabolism concepts. And then I'll work on finishing this up on Thursday, and we'll also be taking a look at cellular respiration and photosynthesis. So my hope is that we can get through cellular respiration this week, and photosynthesis I've over the years just like condensed it, right? Because of the 10% of the things that you will statistically walk away from this class, understanding the nitty gritty of photosynthesis is not high on my priority list. So we're gonna just pare it down, and then if you go take botany or cell bio, then you'll get tortured with all the nitty gritty details of photosynthesis. All right. So let's first introduce metabolism. And again, this is the cycling of energy, and the cycling of energy that's maybe in our food resources that we're consuming to fuel the work that we need to happen inside of our bodies to keep us alive. But it's also, breaking down energy to do work like moving your car, right? We use hydrocarbons like gasoline. And as we break the chemical bonds, right, we release energy that we can use to cause the work to move that car from one position to another, right? So energy is basically a resource for the performing work, right? It's basically you have the capacity to cause change or do a job, right? And we're going to be talking about energy conversions from, kind of a larger scale of the types of energy that we have. So potential energy, just like the name implies, is energy that's stored and available to do work, right? So you have the resources to get a job accomplished, right? Full tank of gas, right, would be high potential energy, right? Potential energy can also be low, right, where you don't have a whole lot of stored energy to do work. Sometimes that energy is going to be stored in chemical energy, right? So chemical energy is the energy that's stored in chemical bonds. And remember, when we break the bond, what we're doing is exciting electrons, and we can harvest that kinetic energy to now perform work, right? And we're going to talk a lot about that chemical energy. And then I've been talking quite a bit about kinetic energy, and that's the energy associated with motion, right? And in biological systems, we're constantly going to be cycling between high and low potential energy, between stored energy and releasing energy, usually in the form of kinetic energy, right? So when you pop an electron out of its orbital, it's exhibiting kinetic energy, the energy associated with motion. And this happens every time that you break a chemical bond. Right. So I'm going to use this analogy quite a bit over the next couple lectures, the idea of storing water behind a dam. Right? What do you guys think? A full reservoir, high or low potential energy?
High.
High potential energy. And then when you open the dam door, right, you release that stored energy, the water rushes out. You're now converting that high potential energy, the stored energy, into kinetic energy, the energy associated with motion. And you can use that kinetic energy and harvest it using maybe a turbine, right, to store electrical energy. Again, we're constantly switching between different forms of energy. Yeah? High potential energy to low potential energy, cycling it back. Chemical energy, the energy stored in bonds, and kinetic energy, the energy associated with motion. So as much physics as you're going to get in here. So I'm going to stop there because I'm at the 10:45 mark. Let me, maybe I have a last poll. Do I have a last poll? I do have a last poll.